Okay, 1 2 1 2. It's fine.
I just want to make sure there's something on here. Sounds okay. No, that's
Okay.
cool.
Okay, good afternoon, everyone. It's just on 2 o'clock, so we're going to get started with the next session. Not sure if there's anyone outside, but if we can just slowly take our seats. So welcome to this session on social ecological systems and resource use. We are in for quite a nice session. Oh, sorry, I shouldn't have said that because I'm talking in the session. We're going to start off with so Alex is not here, unfortunately, so we're going to go straight into a talk by Zabentungwa, who's going to talk about the role of edible insects in rural livelihoods.
Good afternoon, everyone. My name is Zabentungwa, and I will be talking about the role of edible insects in rural livelihoods and identified challenges in Vembe District, Limpopo. Edible insects are an important commodity that is used as a household consumption in rural areas, and the consumption of insects is a common practice in South Africa, particularly in Limpopo and some parts of Mpumalanga, Gauteng, KwaZulu-Natal, and Northwest provinces. They are consumed because they play an important role in nutritious diet. As a result, they are consumed regularly or as a delicacy or to supplement diets during times of hardship when staple food is scarce. Edible insect trade is a good business that generates income and improves rural livelihoods in Limpopo and other rural communities, and the generated income from edible insects varies from species to species. For instance, approximately 16,000 tons of mopane worms are traded per year in Southern Africa, resulting in an income of $59 million a year. And mopane worm traders in Thohoyandou can make up to approximately over 21,000 a year. Edible insect trade could become an excellent commercial business with significant economic returns that can improve rural economy and create job opportunities. And edible insect trade could hugely improve rural livelihoods and create job opportunities. Despite the significant role of edible insects in improving rural livelihoods, there is still no clear policy and legislation that governs the use of edible insects as food or consumption or as trade. There is limited information on policy documents and regulations that support or promote edible insects as food. So the aim of this study was to determine socio-economic benefits of trading insects and their associated reasons for trading insects. And we did this by assessing if edible insects are included in economic development strategies in Vembe District, and we identified economic important species, and we also determined monetary value derived from trading insects. And lastly, we determined challenges associated with trading edible insects. So the study was conducted in December 2019 across 6 towns in Vembe District. Those towns were Elim, Makado, Sibasa, Thohoyandou, Musina, and Biaba. A questionnaire comprising of closed and open-ended questions was used to source information from traders across towns, and traders were selected based on our observations of people buying insects in markets, bus terminals, taxi ranks, and along roadsides. The questionnaires were administered through structural face-to-face interviews with the traders. And we also did an assessment of trading conditions considering food safety, hygiene, trading structure, storage, sanitation, water availability. Table 1 shows demographic information of traders across towns in Vembe District, and 72 traders were surveyed. And we found that majority of traders were females between the ages of 18 to 74. And only 6% males sold edible insects in Vembe District. These results indicate that women play an important role in rural livelihoods, and they are the backbone of subsistence household food security. And edible insects provide an opportunity for women to participate in economic development in rural areas, thus empowering them with income opportunities. and this is directly linked to Sustainable Development Goal 1, 2, and 5 of no poverty, zero hunger, and achieving gender equality and empowerment of all women and girls. And figure 1 shows edible insects that were traded across towns in Vembe district. So we found that 4 insect groups were traded. Among those, mobane worms were the most sold insects across towns. So mobane worms are a popular food source and a delicacy across Southern Africa, and the sale of mobane worm is a lucrative business across Southern Africa. They provide earning opportunities and play a crucial role in reducing poverty in rural areas. These are just the photos of edible insects that were sold in rural area in Vembe district. So figure 2 shows reasons for selling insects in Vembe district, and we found that unemployment was the major reason for selling insects. So unemployment is a major socio-economic issue in South Africa, and this country has the highest unemployment rate. In year 2020, the unemployment rate was 30.1%, and it is increased to 34.9% in 2021, which is alarming, and Limpopo is among the poorest provinces in the country. As a result, people rely on natural resources such as edible insects to generate income and to sustain their livelihoods. Figure 3 shows weekly income generated by traders from selling edible insects. So we found that majority of the traders made between 600 to 1,000 a week, and some traders made above 2,000 rands a week, and on good months during holidays in December or during SASSA grant payments or month end, they said they make double these amounts shown here. So edible insects provide earning opportunities and financial support, and they improve standard of living because the money generated from selling insects is used for paying school fees and paying for basic needs such as electricity and food. Figure 4 shows challenges associated with selling insects. So most traders said the decline in availability of insects was the major reason, and edible insects are collected from the wild, and their availability and the decline in their availability is affected by climate change, uncontrolled harvesting, and habitat destruction. So the decline in availability of edible insects hugely affects rural communities that depend on these resources to generate income. Therefore, edible insects should be farmed to ensure long-term availability and accessibility when natural supply is limited. Figure 5 shows condition of informal markets where edible insects are sold in Vembe district. So we found that the trading conditions were poor with no proper sanitation. As you can see from the pictures, some traders were found sitting on the pavement with no proper protection from the sun and no access to running water. This shows lack of respect and human dignity, which is unconstitutional given Section 10 of the Constitution of South Africa, which states that everyone has inherent dignity and the right to have their dignity respected and protected. Therefore, important cultural practices should be celebrated and embraced by the government, as it's directly linked to Section 30 and 31 of the Constitution. And government could stimulate edible insect business by offering support and infrastructure so that the edible insect business could grow and offer proper storage facilities and funding to improve trading conditions of edible insects. So we reviewed a total of 36 policy documents from various governmental organizations in Limpopo, and we found that edible insects are rarely included in rural development strategies, and only 4 governmental organizations included edible insects in rural development strategies in Limpopo. So therefore, resources and budgets to develop and expand the edible insect sector are limited. And the lack of clear legislation and regulatory guidelines governing trading and the use of edible insects as food in South Africa might be the primary barrier that prevents local authorities from including edible insects in economic development strategies. Edible insects play a vital role in rural food security and livelihoods. As a result, edible insects should be included in food policies and prioritized for consumption. In addition, edible insects should be adopted as rural development strategy that is used to eradicate poverty and improve rural livelihoods and create job opportunities. So we recommend that government and non-governmental organizations should provide infrastructure, farming resources, and financial support to train local people to farm edible insects. This will provide skill development, jobs, and create business opportunities that will improve rural livelihoods. In addition, insect farming will reduce the overharvesting pressure and in the wild population of edible insects. And edible insects can generate a revenue that can contribute to rural economy, thereby reducing poverty in rural areas. Therefore, government officials should invest in expanding the sale of edible insects in Vhembe district because they play an important role in generating income for traders in this district. And I would like to thank the following people for assisting with data collection, and I would also like to thank the following organizations for financial support. Thank you.
Thanks very much, Savanna. Really, really interesting. Are there any questions? We've got a minute or two for questions. Has anybody got anything they'd like to ask? We've got a question over there. I'll bring the mic.
Hello, how easy is it to farm mapani worms?
I think it's easy because they have a short lifespan, so they generate like in large numbers, and it doesn't require much resources.
Any other questions?
I was just wondering about where those mapani worms come from in the villages and towns where you worked and the cost of transport. So do those traders actually go out and harvest themselves, or are they buying from someone? And is that becoming more expensive? Did they say anything about that?
Some of them harvest them from neighboring nature reserves, but most of them, because of the decline in availability, said they buy from the middlemen from Zimbabwe or Botswana, which becomes expensive.
Really interesting. Thanks for that. And I think, yeah, lots of potential for the farming of insects, so it would be nice for someone to do some research on that. I think there's lots of potential. Okay, so we're going to move on to the next speaker, which is me. Okay, thanks very much. So, can you hear me? Can you hear me? Okay. Okay, so I'm going to be talking about the Warburgia program, which I think a lot of you are familiar with, and I've sort of dabbled in it over the last couple of years and flashed a few slides up, but I wanted to do it a little bit more justice this year. Let me use this. I think the first point is that it's certainly not a one-man show. There's a huge team that's working on this project and has been since the very early days. All the names up there are people that have contributed data or designing the contribution of data for what I'm going to present today. Whoa. Okay, and then those names are people that have been part of the program. Whoa, these are not the same. Okay. That have contributed in different ways, in lots of different ways over the last sort of 14 years or so. So very grateful for everyone that's been involved. So for those of you that are not familiar with this tree, this is the Warburgia salutaris, or otherwise known as Shibacha. It's a medium-sized tree. It's an evergreen. It occurs in Limpopo as well as in Mpumalanga and KZN, and a couple of other countries as well. But why it's important for conservationists is because it's on the endangered list, and it's facing very high risk of extinction in the wild. So it's on the red list with Sandby. And this is just a quote from some very early people documenting it. So they are all over the country, only poor coppices, every year cut right down to the bottom and used all over and sold. So that was a report from 1938. So this is nothing new. You know, these plants have been harvested and being over-harvested for a very long time. It's a very popular plant. It's ranked as the first or the second most popular plant with many traders, traditional medicine traders. So that, of course, is an important acknowledgement. So the reason it's important is because of its bark. The bark is what's used primarily for traditional medicine. It's used for colds and flu, mainly. It's a very successful expectorant, so something that you might recognize from the pharmacy, such as mucospec, would do the same sort of thing. And it actually is used for a number of different things, as well as more recently used to treat corona. It gets sold in the form of these, in sort of bundles of bark on the markets, and then it gets used in the powdered or ground up form. So that's mucospec. It does the same job as mucospec, and it's popular because it works. What's sort of relevant to Kruger is that we've got one of the largest remaining populations of Warburgia in the wild, and it was being illegally harvested for a long time now, but the way in which the harvesting has taken place changed sort of towards the beginning of the 2000s. So you can see quite a difference in the way in which these two trees have been harvested. Much less sort of sustainable approach on the right. So that was obviously a cause for concern, especially when some of these sort of things were being recorded. These are trees inside Kruger, massive trees that were being illegally cut down. Unfortunately, they do occur quite close to our western boundary, so it makes it quite vulnerable. And of course, what happens when you ring bark, then you'll get crown death as well. They're quite prone to coppicing, so they're quite a robust tree, but of course they do eventually die if they are harvested to an extreme sort of level. So this led us to start the Warburgia Conservation Program in 2009. The idea was to promote and facilitate the conservation of this plant. And we started off by putting rangers in the field. That was the first sort of point. Put guys out there, we knew where the plants occurred, and they basically were protecting the plants for us. We also decided to look at, do some monitoring and make sure that we were aware of what the population was doing, and do some research around the reproductive ecology of the plants, and Dave's going to talk a bit about that after me. But then the component that I've been most involved in is this third objective, which is to do with promoting sustainable use of Warburgia by means of promoting access to alternative sources of the tissue. So I'm going to talk mostly about that. So the first two objectives we started in 2009, and then we started the traditional healer work in 2010. So what it really started with was a whole lot of conversations with traditional healers, right up and down the park, finding out what are some of the concerns of the healers. We met with lots of traditional healers. We chatted to them in different places. We met up at traditional healer nurseries, such as this one in Tulamahash, and we just discussed ways in which we could work together to try and develop this program in a way that it would have beneficial outcomes both for people as well as for conservation. This is a photo that we took from a group that we met up in Venda. So we've covered quite a wide range of people in the early days. So the basic point was that there was a lot of interest, there was positive interest, people wanted to participate and they were ready. So we started trying to grow these plants. The idea was that we'd grow plants, distribute them, make them available to healers who would then grow them at home, and they wouldn't have to then buy from the markets that were accessing, the traders of which were accessing illegally harvested plant tissue. But this proved quite tricky. A lot of our saplings died. This was in 2010. But in the interim, we made leaves available, just to buffer the use in the interim so that people could have tissue that was harvested sustainably, but then not actually support the markets. So we made a whole lot of leaves available to traditional healers. We packed them in little bags, and that was sort of the start of it. But by 2012, we still had no little saplings. It took a long time to get going. So we actually ended up buying our first batch of 40 from a little nursery down in Komatipoort. And we very selectively distributed those. They were very special to us in those days, to some strategic traditional healers that we knew that were living in the areas much closer to where the trees occurred naturally in the park. But then by 2012, things were looking pretty good. And we literally now have thousands and thousands of these plants that we are able to use in our distribution program. This is thanks to a team led by Michelle Hofmeyer, who certainly sorted out the propagation issues with those plants. And we now literally have loads of Warburgia. So that, of course, opened lots of opportunities for us to now engage at a much broader level with traditional healers. And we did this over a number of years, having discussions mostly about the availability of species. So what sort of species do people find difficult to get hold of? The use of different plant parts. Most people traditionally would have used bark for Warburgia, but we were interested in people's feelings around using leaves because it's more sustainable to harvest. Thoughts on nursery propagated plants was quite important. And then just discussing general challenges of people, acknowledging that traditional healers in our context are a really important group within society, holders of important institutional knowledge as well as stabilizers within the community structures. And it was really important for us to have positive relationships with these wonderful people. So at the workshops, we also did a planting demonstration with the healers, showed them how to plant some of these plants and how you would do so in a rural sort of setting. And then we also used the workshops as an opportunity to select representatives of within the villages. So each village had a traditional healer representative who was then our contact person for the healers that were not at the workshops. We couldn't engage with everyone. And then also just to have a contact to do follow-ups with the projects. And then we used the workshops to distribute trees. So distributed literally thousands and thousands of trees over I don't know, it's probably about 360 kilometers from north to south, the geographical area. And all the healers that participated went home with a couple of Warburgia, as well as enough to provide the healers that were not at the workshops within their villages. And this was a very sort of successful way of getting these plants out. So each workshop we held had about 20 people, representing between 2 and 12 villages, depending on the area. We've held 20 workshops between 2010 and 2018. We've engaged directly in this manner with 400 traditional healers, and then through their frameworks with another 2,500 traditional healers. We've distributed trees to over 100 villages next to Kruger, and we've distributed over 30,000 of these saplings now. Just want to note that of those 30,000, not all of them have gone to traditional healers. We've, of course, we've got so many now, there's almost not enough healers for the trees that we've got, so we've been distributing to other user groups as well. So if you can see there, not a good idea to use green, but each one of those little spots there is a village that we've engaged directly through this program. So you can see we've done all the villages next to Kruger, and each village has got between sort of 400 and 300 traditional healers, and each traditional healer has got between 1 and 7 Warburgia. Just to give an idea of the scale and how it's changed over time. So those were the first little 40 that we bought in 2012, and we distribute now on average about between 4,000 and 5,000 trees annually. And then we also speak to them about different other species that they find hard to get hold of, and this then has guided the development of different species that we're now distributing as well. So we go back every now and again when we can, and we visit people, have a look at how the trees are growing, and had really, really nice feedback. It's really nice to go back and see people that have been part of these programs. But this made us sort of ask some more questions. There was a lot more that we actually needed to know. It wasn't good enough just to go and look and say, okay, that's great, and take a photo. So what were the survival rates of these donated saplings? We predicted that they'd be pretty good from what we had seen. What were the growth rates and the phenology like of these saplings? We predicted it would be pretty high next to the area where they naturally occur. We were interested in whether the saplings were actually being used. So are we distributing all this stuff and people are still buying on the market? That was quite important. We predicted that they would be being used. And what were the sentiments of people? They had said at the workshops that they were happy to use nursery-grown plants, but was this actually the case? And what plant parts were being used? People said they were happy to use leaves, but are they actually using leaves? And then what does this mean in the bigger picture in terms of supply and demand? And we had some predictions around that people would no longer be buying from the markets. So recently we've done some more systematic monitoring visits. You can see how beautiful some of these trees look over here. These are taken this year, where we've actually gone and visited people and done more detailed measurements. So firstly we record survival, so we note how many saplings people got, a traditional healer got versus how many survived and why they may have died. We measure stem diameter or circumference depending on the size of the plant, height, as well as some leaf canopy measurements because the amount of leaves out there was quite important to us. And then we also take note of phenology, whether they've fruited and flowered. We also look at bark harvesting. So are people actually harvesting bark? Most people are harvesting leaves from their trees. Are they harvesting just the little stems like this one, or are people actually harvesting from the main stem? And then we have conversations around things and just see how the project is actually working. And this can either be done on an individual basis or with a group, depending on the village context. And then we talk about the usefulness of the plants, what parts are being used, what volumes are people using, do they have enough, what is the source of material that they're currently using, acceptability of garden-grown plants, and then any other issues that might be relevant. So what I want to point out now is that this was never set up to be an experiment, so it's not a perfect kind of research design. We started distributing up there because that's where the trees grow, and we kind of worked our way down based on risk. So when I'm giving feedback now of the sample, just bear that in mind. We don't, as social scientists, care about that stuff, but I just put that in for you guys. So my sample is covering, so the data that I'm going to present is covering that area, and those dates over there are where those people received those trees, okay? So the current sample is 99 trees, not bad for a social scientist, hey? But it's just the beginning, so we're just sort of starting off, and that's kind of the breakdown. The trees come from 35 different households and represent 28 different villages at this stage. So just a note on the traditional healers that we've interviewed up until now. So most traditional healers that we've interviewed are female. Most of them are above the age of 60. The average age of qualification when people became traditional healers was 33 years, and the average number of years that people have been qualified for was 25. Just interesting to point out, the person who qualified at the youngest age of the interviewees was 14. And we also interviewed somebody of 81 years old who is still very practicing quite regularly. So look at that survival rate. I think that's quite awesome. Well, I did until I showed it to Tony, and he was like, not bad. I was expecting it to be better, but I think that's quite good. We have had some deaths. Just out of interest, of the ones that did die, 62% of them came from one single cohort. And I do remember that cohort, and it was one that sat in bags for quite a long time. And I do think that might have influenced the survival because they died pretty quickly. Just out of interest, this was the only kind of biggish tree that we were able to record that did die. So it seems they either die very soon or they don't die. And this is this one over here that actually seemed to have died from having too much water. It was sort of planted, there was a lot of water about a year ago, there was a lot of rain, and it seemed to have got waterlogged there. So this is just to show one of the ladies who got one of those very first 40 trees over there, receiving it in 2012, and this is what her tree looks like now. There she is there. She is tiny, but that is a big tree, okay? Thanks, Tony, for the graph. I have to have one graph in every talk. Here it is. This was just to show the growth rates. This is months since planting and the height of the trees. You'll be quite interested in that little one over there. Just to show that we haven't really seen too much difference in terms of geographical distribution, but we're going to increase the sample size and then see if rainfall might be playing a role. There's obviously a lot of other things that will play a role in terms of how quickly a plant is going to grow in a garden, including harvesting. But this is a very special tree over here, so you can see it over there. It's a monster. I think we measured it at, I think it was about 16 meters tall. And this was actually a sapling that was given to a traditional healer in 1996. We have since engaged with her granddaughter as part of our program, and she's since passed away. But she was part of a group of traditional healers that were here, invited into Kruger in 1996 with the opening of the Tulamela archaeological site. And they were then given a couple of saplings. So we managed to find this out there, and it's been out there for 26 years. So it's a planted plant, which is really cool in terms of us then being able to predict future growth rates and so on of these trees. So we know that trees are being used and that they are really helpful. All respondents are using the trees and finding them helpful, and 80% actually indicated that they used them during and for COVID. which was really useful. So I've healed a lot of people using these plants. It helps a lot. And during COVID, lots of people came from around to use the leaves. So lots of leaf use happening. You can see the majority of people are using leaves. Lots of healers using leaves and bark as well, but very few that are actually harvesting the roots. And many people have actually said that they won't harvest the roots until the trees are a lot bigger. What's quite interesting that some people do treat different ailments with different parts of the tree. So just in terms of the impact on the local market, quite important for us to then see if people are still buying or selling. Currently, nobody is buying any material. They are only using the plants that they harvest in their garden. 89% of people used to buy, that we interviewed, so most people used to buy the plants. A couple of people have actually started selling tissue material, so there's people that are selling bags of leaves that they either sell fresh or dry, and then there's actually people that are propagating from their plants that they got in 2012. So there's currently a person that we know of, one of our traditional healers, that has got 500 little saplings in his garden that he's propagated from his own plant and that he's selling on to people in Venda and so on. Most people have said that they do have enough. Some want some more, mostly for others. There's always new people qualifying as traditional healers, so to be able to supply everybody. And quite a lot of people do donate material, and it's mostly to other healers that don't have. Just out of interest, don't worry too much, but I think this just is where people used to buy their material from before they got the plants in their gardens. So a lot from Burgers Fort, but I think this is quite interesting. So the second most highest place where people used to get their material from was from the Kruger population. So just in terms of homegrown and wild harvested plants, everybody's happy to use garden plants, and they've said that the actual plant is the same. The concerns were more about pollution. So not pollution in the sense that you or I might understand, but it's more pollution from coming in contact with people who may be in a condition that would result in a negative impact on the ability of the medicine to work properly. So these are people that may have attended a funeral, or perhaps a woman that's menstruating, or somebody that's been intimate, for example. These are all things that can result in the medicine not working. So what people have actually done, there are processes and rituals that can actually counter the pollution process, but then a lot of people are actually fencing their plants off in their homesteads. So it hasn't proved to be a problem because people are able to manage, the healers are able to manage the potential negative impacts. You can see there's a lady from Hlalagahle who's got hers all fenced off at the back, and nobody's actually allowed to go in there except her when she's harvesting the plants. And there's a couple of examples. This is another example around Palabora area where they've fenced off the plants. Just in terms of the supply and demand, we thought it was quite, let me flick back there, just quite useful to say, well, okay, there's these literally 30,000 trees that we've now given out. What does this mean in terms of the bigger market? What about the bigger picture? Are we actually having a dent in this? So Tony helped me to run some Beckful analyses on the leaf canopies of the trees that we've distributed. A lot of sort of assumptions in this table, but I think the point is that currently there's the approximate leaf mass that is out there is about 22, just nearly 23 tons. So that's based on the distributions that we've got. That's leaf mass, dry leaf mass, not bark. So that's quite a lot, we thought. So then I delved a little bit into the literature to see, well, what do we know about the commercial trade of Warburgia? And this is the only data that we really have. So in KZN in 1988, it looked like they were using about 15 tons. And then in the sort of Gauteng area, 41 tons. And then in 2001, there was a study on the Faraday market, but that's a subcomponent of that. So if you add those two together, you get 57 tons. So the leaves that are currently out there would supply just still quite a small fraction of that. So this is just quite useful in terms of how we're looking at this bigger picture. We, of course, are able to support the subsistence market, as we've seen, but we really need to get into the commercial markets and see how we can try and reduce the illegal harvesting from the wild so that we can promote access for some of our healers into that market. TOPS is a massive problem, the permitting process.
So that's proving a huge bottleneck, but we are experimenting now with wild ginger to try and look at ways in which we can legalize some of this stuff so that people can actually legally enter the market with their produce. So this is wild ginger, distributed quite a few of those already. A very different plant, which will have very different outcomes and challenges in terms of harvesting, because the tuber, the rhizome is what's harvested. But there's already people growing it and using it within their households as well, and on a commercial level. So this is two different commercial farmers of wild ginger, and it might actually more be a case of us supporting the commercial farmers and then providing the healers, as opposed to the healers growing the wild ginger themselves. So we've been helping these commercial growers to be permitted, so that we can actually legally buy from them and then donate those plants to the healers. We've also supported our healers during COVID. I won't go into that. And we're also exploring other opportunities for supporting traditional healers in ways that we can. Just out of interest, so elephant dung is widely used to treat a lot of ailments, and the demand at the moment for the traditional healers next to Kruger would be somewhere in the region of 6 tons, and that's how much we've got. So we can definitely share some. Okay, so just to finish off, I think the sort of main message is that all this engagement and all of this work, it's not just about that, it's not just about Warburgia, it's actually about relationships and building and maintaining relationships with a really important group within society. And there's a lot of different ways to do that. But if you are going to do that, you've got to do it regularly, and that of course takes resources and people to do. So the other point is that this is not a blueprint, but I think there are very real opportunities for us to be able to engage in some of this stuff more broadly. Each individual species that you look into has got its own context, and that's why these processes have to be very flexible, and you have to be able to learn together with people as you move forward. I've put this picture in. It's from Dave, who's going to be talking next, Dave Thompson. And the point here is that it's an ongoing journey, because this is bark harvesting that took place in Kruger within the last, I think it's about 18 months. So it's a continual process, and it's not like we'll be able to tick the box ever, but we're still going to keep going. The other important thing to remember is that, or just to note, is that Warburgia has now been downlisted. So it's no longer listed on CITES as on the red list as endangered. It's now vulnerable. Okay, so thanks very much for listening. I know I went a little bit extra time. I stole some from Alex, who wasn't here. I'm sure he wouldn't have minded. But just want to thank the Honorary Rangers specifically, because they've helped us a lot over the last 10 years in various ways, and also supported quite a lot of us to attend this conference from Sandparks, which we wouldn't have necessarily been able to do. Thank you very much. Where is Dave? There's Dave. Okay, so we're going to move on to Dave. I'm not going to ask questions because I'm afraid of you. It's just because I feel bad because I talked so long. Okay, Dave also wants to steal 2 minutes. So far we're on 7. We're on credit, hey? We've got 7 of Alex's. And he also talks long, so we've probably got a bit extra. Okay, so Dave's going to be talking about the reproductive ecology and genetic diversity of the pepperbark. Thanks, Dave.
Right, thanks Louise, and good afternoon everyone. I think Louise has done a fantastic job introducing the species, so I'm not going to talk too much about that. And Louise also spoke about this fantastic propagation program. But what I would like to do is raise the flag and say we need to be concerned about the species persistence in Kruger despite the propagation program. So we're going to unpack that a little bit. First of all, I just want to thank my co-workers, Kelly van den Bosch, Kelsey Glennon, Glennis Cron, and Edward Koske at WITS, and Jacques Leroux now at Macquarie University. Also just acknowledge the funders, SANBI, FPIP program, the Botanical Society, and then also the NRF for student support. Okay, so of course we're talking about Warburgia salutaris, it's critically endangered, blah, blah, blah. Louise has told us all about that. But at this stage, the distribution is really southern Mozambique and northeastern South Africa. Sorry, thanks. And as Louise had said, it's primarily the harvesting of bark, which has driven the kind of the status of the species as we have it at the moment. South African context, it's very popular in local communities in terms of their healthcare. Restricted primarily to protected areas, including Kruger, as has been mentioned, they're right in the north. But considerable local extirpation. If you look at previous distribution, the crosses versus current distribution, you can see there's been a lot of loss of populations in the last 20, 30 years. And the species responds very well to damage. It responds very vigorously with coppice, and the bark recovers relatively easily. And that, to me, is artificially supporting these populations in terms of the number of stems that we see and in terms of their ability to produce bark that can be used. So the Kruger context, right up in the northwest, there's 13 subpopulations. Those range in size from one individual up to an estimated several hundred. As Louise mentioned, they're very close to the border, at most 4.5 kilometers from the western boundary. And because of that, there is the ongoing bark poaching. We've also heard about the Sandparks program, which had sort of 3 pillars: to multiply, protect, and monitor. So I'm not going to sort of unpack those. I mean, obviously, Louise spoke about the fantastic success of the multiply program. But we're going to sort of expand on the monitoring aspect. And what we've noticed in the field and what early researchers found, firstly, is that the size class distribution of the population is normal. It's not inverse J-shaped, as we might have hoped for a sort of sustainable population. And so there's evidence of a lack of recent recruitment. Flowering is sporadic at best. Fruit is hardly ever seen in the field, and when you do find it, it's parasitized. And then there is evidence of vegetative regeneration. Reports of clonality from KZN and some infield observations from Kruger of trees being connected to one another. And so that really gets to the crux of this talk, is that you have this kind of antagonism between asexual function and sexual function. And so what we're going to do is unpack that and have a look at the prevalence of clonality in the Kruger population, in terms of genetic structure and diversity, and then also have a look at some of the reasons behind the failed sexual recruitment that's been reported over the last couple of years. And then really just think about the implications of this for the recruitment and the persistence of the species in Kruger. So in terms of the genetics, that's the 13 subpopulations in Kruger. We sampled from Kruger 227 individuals. We collected leaves from within Kruger, and we included 31 nursery-grown plants. Now, bear in mind, we started this back in 2012, I think, and so this was before the multiplication program had taken off, so we didn't have a lot of nursery stock to work with, but we had included 31 nursery plants from 7 different nurseries. And just to kind of clarify, we collected material from what we thought at the time were individuals, because they were not obviously connected to each other, and they were several meters apart from each other. And then, of course, there's the stock material outside the park. Very quickly, we developed some polymorphic satellite markers, microsatellite markers, to have a look at the genetics. The first thing is looking at the clonal reproduction using POP-R, tests for linkage disequilibrium. This is in the wild samples. Okay, in a naturally reproducing system, you would expect to be here on the blue line. We've got the significant deviation away from expected. Okay, that gives us support for what we call linked loci. Okay, so immediately there was evidence of prevalent clonal reproduction in the Kruger population. Okay, if we have a look at the genetic distribution, like I said, we sampled 227 stems in Kruger. We only found 113 genotypes. Okay, which means that half of the stems share the genotype with at least one other stem. At least half the population is clones. Just a couple of examples. That subpopulation, it only had 14 stems. We sampled them all. It's a single clone. On the other extreme, we had a fairly large population. We sampled 31 stems, mostly unique individuals. And the largest population we worked in Kruger, we sampled around 100 stems. 75% of the stems share their genotype with at least one other stem. Okay, so most of the population is comprising of clones. In terms of the stock material, we found about 35% of the stems were clonal. Okay, so if you have a look at this in the field, that's a map of that large population. It's 100 or so stems. But where it gets really fun is if you turn the stems into clones. Okay, so instead of having 100 individuals that could be reproducing, you have around 25 clones. And the important part about that is that they can't reproduce with themselves, and we'll get to that in a second. Okay, in terms of genetic structure, the population, or the Kruger population at least, has got around 11, or has 11 genetic clusters within it, and those correlate strongly with the different subpopulations, and the larger subpopulations break into smaller clusters by genotype. Okay, so there is very little gene transfer happening between the subpopulations in Kruger. That was supported by principal coordinate analysis. The Kruger populations separating out here in genetic space, okay, all of them separate individually. And then what's important to realize or note from here is that the stock material, the material that's currently in cultivation in the micropropagation program, is genetically distinct from the Kruger material. Okay, just some diversity metrics. I'm not going to spend a lot of time on this, but just to show, and it's kind of paradoxical, you wouldn't expect it, but here's some big subpopulations in Kruger. The genetic diversity is really high, and the same for the stock plants in the nurseries. Genetic diversity is really high. And just to show that that's actually right, there's our population that comprised just one clone. Okay, there's no genetic diversity there. Okay. Right, in terms of reproductive ecology, we focused our attention on the 4 largest subpopulations in Kruger, and then one small subpopulation where old fruit had been found a couple of years previous. Flowering work was done May through June. It's a winter flowering plant, and the fruiting work was done November, December, the subsequent summer. All right, in terms of the breeding system, over 2 years and hundreds of plants, we only managed to turn up 42 flowering stems. Okay, and what we did with those stems, we conducted a range of pollination treatments, autonomous selfing, guide-monogamous selfing, outcrossing between subpopulations, and then allowing for natural pollination with and without protection from seed predators. Okay, right off the bat, we found that it is not possible for the plant to autonomously self. It cannot cross-pollen within the same flower, within the same individual, and produce viable seed. But even so, the other pollination treatments that we carried out, the outcrossing and even the natural pollinations, had very low percentages of fruit initiation, okay, at most 16%. Okay, so low fruit initiation and even lower fruit maturation. Only 2.3% of fruit that initiated went on to mature. So we've got a population in Kruger which is characterized by very low fertilization and very high fruit abortion. Just having a look, amazingly, this had never been documented before, but the pollination syndrome of the species, lots of visitors, Lepidoptera, Diptera, Coleoptera, Hymenoptera, and Thytanoptera, the main ones. But based on the abundance of what the individuals we found, the contact that they would possibly make with the reproductive structures, and the pollen loads that we were carrying, we are putting forward the moths and the butterflies as the likely pollinators of the species. Looking at the male function, just looking at the viability of collected pollen, depending on the subpopulation, anywhere from 66 to 93% viable pollen. And irrespective of how we cross the pollen between pollen donor and recipient, we had about 32% pollen tube growth, but only about 7% of those tubes actually reached down to the ovules. Pollen production was not great, I'll be honest. It was relatively low, but not sufficiently low to sort of account for the low fecundity that we're seeing. And in one subpopulation, we encountered what we later identified as failed anther dehiscence. Okay, the anthers were not opening to release the pollen, and that is a documented genetic or form of documented genetic male sterility. All right, so fruit set. So hundreds of trees, 42 flowering individuals with hundreds of flowers. and just 3 fruiting individuals. 2 individuals here fruited in 2019. They 2 stems of the same clone, and then in 2020 an additional stem with fruit was found from a separate clone. Okay, a lot of the fruit, 9% have sort of some damage to the base of the ovary, which likely results in ovary abortion. About 20% of the fruit are parasitized by the marula fly. But what's really trying to asexual vegetative growth, and this would be an example or indicative of a population that is currently in disequilibrium with its habitat. So we have what we're saying is a relatively recent failure of sexual function of the species in Kruger, and that at this stage the species is persisting entirely via clonal reproduction, and that is linked probably to a recent or fairly recent environmental shift, but we're also considering the link with human use as a possible driver of the abandonment of sexual function. And unfortunately, the trajectory of the population is such that there is likely to be further lost reproductive capacity as a result of genetic depression and inbreeding. Okay, so what can managers do? Tiered sandbox program, the multiply, protect, and monitor. Okay, in terms of multiply, okay, we've heard there's what do you say, 30,000 trees in circulation, diverse, but they are distinct from the Kruger genotypes. So the priority needs to be, and the little seedlings that we produced last, or the year before last, have filled that gap somewhat. And with the information we now have about the clones, we can do some informed searches for flowering individuals and fruiting individuals, and carry out informed pollinations and bagging experiments to try and maximize the number of fruit that we get. In terms of protection, we need to think beyond just protecting the trees in terms of bark harvesting. We also need to think about protecting their genetic diversity. In terms of monitoring, we really need to reassess the Kruger population. We need to think in terms of the effective population we've got, how many genotypes we've got, rather than the population we thought we had based on the number of stems. We need to take what we thought we knew about the size class distributions and the expectations around that and throw them out the window, because this is not a population that's going to behave in a way that we currently understand. There are also pre- and post-emergent aspects in the reproductive cycle that we need to concentrate on. It's not just about developing the seed, but also aspects beyond the production of the seed and the seedling. And I think there's a lot to learn from comparing to the populations outside of KNP. And then my last point is kind of a challenge to rethink. Okay, for an animal that we're reporting for Werburgia, people would be paying much more attention than they currently are. We probably need to acknowledge that in Kruger, the species has a defined endpoint, okay? Even with protecting from bark harvesting and producing or putting trees into circulation to protect from further bark harvesting, it's likely that the population has a defined endpoint. We need to go beyond the tree being a medicinal resource. Louise said it's not just about the plant. I'm going to say it's not just about the bark. This species deserves our attention for reasons other than the fact that it's heavily utilized. And then obviously there's a lot that we are just starting to get our teeth into here, and so we need to prioritize and continue with the research that we've started to try and better understand why the Kruger population is the anomaly, or what we think might be the
population. And you're talking about the Kruger genotype, but are those trees not supposed to be actually outcrossing with trees further afield? So what happens if you pollinate them with pollen that comes outside of Kruger?
Right. So that's the one thing that we would want to do, is we would want to do some guided pollinations using pollen from multiple sources, not just from within Kruger. But if we think back to the PCA plot, the material that's in the nurseries, which was derived initially from KZN plants, is distinct genetically from the Kruger material. So we may not expect that to produce viable seed either. It may have been fairly limited in its ability to share genes initially anyway, even though it was continuous. It could be as well that the distribution has been kind of disjunct for a very long time, and that populations equally so, Kruger and elsewhere, are all feeling the effects of kind of their genetic isolation.
Thanks, Dave. I also wonder about those populations in Kruger. You know, if we look at the size classes, there's only a couple of monsters and the rest are little. And you do wonder, I mean, it's a very sort of historical area where people have lived for a long time. So it might also be that maybe, you know, maybe they weren't meant to, you know, maybe they were moved there, like we're sort of thinking for the Savanna. So they would be quite nice to look at. And those big ones, you know, the ones that are now, I actually can't believe I forgot to include the phenology stuff that we've, but there's a lot flowering and fruiting outside from that KZN stock. So that might be quite nice to look at to eliminate some of those possible variables up in the north.
So maybe just to add there, so the success of the program that Louise was talking about is rooted in seed that's sourced from an orchard of Warburgia plants that grows in KZN. Okay, it produces prolific fruit. When you walk through there, it's like walking in an olive orchard. The trees are literally hanging on the ground with fruit. So this is not a problem, this lack of reproduction is not a problem.
The dominant genera in this forest types was the Hyperponera and the Parasitiae. These species mainly nest on decomposing wood, soil, and leaf litter. The high diversity in the eucalyptus plantations was potentially due to the presence of logs, which may have diversified nesting and feeding grounds, but of course there are other factors that could contribute to this. As we have seen, ant species richness and abundance did not differ. However, diversity and assemblage composition did vary. Many species were shared between the habitat types. This implies that a simple habitat type does not necessarily result in fewer species. Therefore, we can conclude that the eucalyptus plantations have the potential to conserve ant taxonomic diversity, given that they are planted close to a natural habitat. Recommendations for future studies include investigating ant functional diversities, as these organisms are ecosystem engineers. Furthermore, specific environmental variables such as soil properties and temperatures have very important roles and may have contributed to such outcomes. The effect of patch size and forest age, as well as edge effect, could also contribute. I would like to thank everyone who participated in this project and my project funders. Thank you for listening.
Thanks, Thembekile. So we can have one quick question if there is anything. No, nothing. Thanks very much. And if they have questions, I'll grab you at the poster session. So we're going to be moving right on. Our next speaker is Caswell, and he's going to be talking about ant biodiversity in KwaZulu-Natal. Thank you.
Okay, so understanding where biodiversity is and how it's changing over time in space is very important. Similarly, we need to know what drives that biodiversity, what drives the change, and what is the response of the ecosystem services, and then what are the consequences of that to us as human beings for some selfish reasons, right? And also, what are the options available for us to sort of respond to such changes, right? So I studied this group of organisms that I'll talk about shortly. So in KwaZulu-Natal, mostly you know us for the blue gum plantations that Thembi was talking about just now, and also the sugar plantations, stuff like that. As well as there's a lot of research done, of course, in the charismatic animals, very beautiful animals, as not Tom, but Dave Thompson has been talking about. But little is done, of course, in terms of the majority of the organisms which are out there, which are the invertebrates. So on behalf of all the invertebrates, so we study these ants in my lab, and I want to spend the next 3 minutes and a half, talking about some work that my lab does for a living. Okay. So here's some of the studies. So I'll start right here. Because I want to sort of show you what we've been doing in the past 3 to 4 years. So starting from some of these bizarre ants called the subterranean ants that, of course, subterranean is, of course, they are crawling underneath the soil, and they are out there, which we did publish at some point in the same study site that TMB was just presenting now. Okay, so these are the kind of stuff that I'm talking about, which are very diverse, but in South Africa, in Africa across, there's been 2 studies that is look at this. It's that one of ours, and then another one done in Cameroon, and that's it. And we don't know anything about anything more than that. Okay, so we are so lucky in the University of KwaZulu-Natal, we host one of the oldest fire experiment. And one of my master's students who just finished recently, she did look at how ants respond to these trials of fertilizers and burning, long-term burning. I think this long-term burning now is 69 years old. So we did, she found out some very cool stuff here regarding the response of these systems to, the response of these, of ants to these disturbance. Another study that is not sort of checking is what we're currently trying to publish, which looks at the expansion of all these woody plants into the open habitats, which have got detrimental effects for biodiversity. And it doesn't matter, even at the vertebrate level, we've seen most studies showing that this can be very problematic. Okay, so these are some work we did previously. Of course, similarly, we have got very much sensitive habitat types in the province, such as this sand forest here, which, yeah, we are lucky that we've been studying those as well and sort of checking how diversity and assemblage changes between these habitat types. These are the woodlands which are sort of in ecotone between the savannas and then and this sand forest. Okay, like I said, I'm giving you highlights of my studies. There's 2 minutes left there of many studies. There's an ongoing study as well that we're showing that we're doing with one of my PhD students looking at how arthropods and also some of these the grass and forbs sort of respond to land use changes such as your human settled environment as well. Okay, one notable work as well is what's already been completed by my former master student who looked at the benefit of this reforestation, okay, and how the progress of reforestation using ants to measure those type of to measure the progress of the reforestation reforestation. 2 more studies that we're doing are looking at the response, or that we did previously that we were trying to publish, focused on the response of these ants to invasive alien species. The most common one in the province is this one, and like just like anywhere else, in Malanga, Komolino, or Darata, wherein we study this along an invasion gradient. So, and also recently, just like Llewellyn is doing, we're not copying from him. I think we started first. We're also looking at how ants are responding to this other plant called Parthenium as well, which is very much problematic. This is an ongoing study now. Like I said, I'm just giving you highlights of what we're doing, and I've got 5 minutes there. I've got 1 left, right? So the last one, or second last one, is this study where we look at, because many things drives ants. Things like competition are very, very important. So we did look at the role of things like competition in structuring the grassland and communities, because majority of our province is actually grassland. And we did highlight in this study that is already completed and is currently being prepared for publication as well, on how ants sort of, how competition structure these particular organisms. Lastly, is this crazy ant that my student just submitted now, which is trying to, this is supposed to be one species, but it looks like there's many, many other ones. Of course, just like any other invertebrates out there. This is one of the problematic ants almost across the country in terms of just being a junk ant. So we're trying to redo the taxonomy of this. This is work submitted and it will be prepared shortly. Like I said, we don't have much more time, so I just give you some highlight of what we do, and thank you very much for the little time you give me.
Thanks, Caswell. You used your time very well. So we're going to move on to the last talk in the session by Ludzulu, and we're still staying with ground-dwelling anthropods. Thank you.
Good afternoon, everyone. I'm still waiting for the presentation to project. Yeah, as you said, I'm Ludzula Mkwebo, so I will be presenting to you the impact of long-term prescribed fires on ground-dwelling arthropods at Kruger National Park. So just to give a background of where it all started, fire is known to be amongst, significant ecosystem drivers, which, has a significant effect in flora and fauna in the ecosystems. So it is still as well used as, management strategy or practice in various landscapes. And as well, there's been greater concerns of fires that, goes beyond demarcated landscapes, in which are incidental fires. So for these reasons, there's been some policies developed in order to manage, such an act in which, the policy were gazetted sometime in 1948, and post that, there's been, long-term fire bent, experimental bent plots, which were established, as Caswell say, it means it's 3 years after they established theirs at Ukulinga. So there's been a growing interest on the benefits that fire may have in various ecosystems. Right, these experimental bent plots were established with an interest of trying to assess the overall benefits that the fire may have in terms of, flora, fauna, as well as carbon dynamics. For the purpose of, my study, I intend to at least document the effect, the long-term effect of these prescribed bans on arthropod diversity. So just a brief on the study site. The study was conducted at Kruger National Park, in which, there is one site at the southern part, which is a Skukuza area, and the other site is at Mupani site. And, pitfall traps were used as well, in order to optimize capturing, efforts. Then it was as well supplemented by active searches. So in terms of the abundance of arthropods which were captured, we captured the overall 6,765. At least I saw the earlier speaker presenting 300 and something, which make me to feel better anyway, because I've captured a whole lot. And, most of those were captured at the unbent plot, hence the lowest number was captured at the annual plot. But when it comes to the overall number of species captured, most species were captured at the annual bent plots. And after having to run some estimators, we realized that irrespective of the number that we have captured, we have not done enough, which is a common thing in ecological studies. Then on the abundance, as the previous table highlighted, the mean captured per plot, we had the lowest at the annual bent plot, hence there is no significant difference between the triennial bent and the unbent plot. But when it comes to the overall species richness, then the results were opposite, where in the bent plots we had a whole lot of, arthropod species captured. Just to give, the likely figures or reflection of what has been captured, a whole lot, of organisms, they were within the group of ants, in which it constituted at least, about 76.4% of the overall, arthropods captured. Hence, we had, beetles at 18.6 and other arthropods thereafter. In terms of, the overall species which were captured, we realized that, as much as beetles were not the most abundant, but in terms of the species richness, they are well distributed. Hence, ants are at a second position. So further to that, I think something may be wrong. My figures are not shown clearly, but I'll try to explain with the best of my ability. So for all groups of arthropods, in which looking at the four major groups of arthropods which were collected, ants were the ones that sort of responded to this kind of disturbances. And then the same data was documented for the species richness. In terms of diversity, none of the group independently showed an element of diversity, hence multitaxa did show some diversity. So in terms of shared species or species similarities, the classification tree and as well as MTS coordination showed that most species are shared between the triennial and annual, in which those are our disturbed plots. Hence, the unbent, is sort of, distant from, the rest. So in conclusion, I would say, annual fires, they threatened, the overall abundance of the species which were recorded. But, as well, this type of disturbances encourage the overall richness of the species which were sampled. And then the most abundant group has had a significant contribution or influence towards, the overall results that, I just presented earlier. Personally, I thought, I should raise this last one to say the triennial bands encourages, conservation of arthropods, since, there are, sort of more species, richness as well as, the abundance is fairly high compared to the undisturbed area. I would like to thank the two institutions for financial support, the two for administration or coordination of the research at Kruger. Hence, the other two members are thanked for field assistance, and Tatenda for some statistical assistance. Thanks.
Thanks, Ludzulu. They will get you at the poster session if there's any questions. So now we're going to be shifting gears a little bit, and we're going to go to some wildlife sampling methods and insights from novel technology or techniques. And our first speaker is Terry Lee, and she's going to be speaking about bycatch. Thank you.
Thank you. Cool. So, I'm Terry. So one of the major considerations when managing biodiversity is knowing what you're managing and at what intensity. But how do we decide which aspects need more attention? Do we just leave a system to manage itself? Well, with the use of fences to protect conservation areas, this isn't really an option. We now require a certain level of management intensity. And as we've heard over the last few days, there's a lot of different aspects to consider in this, everything from soil to people and all of the aspects in between. But I'm going to focus on carnivores. So as we know, certain species often get the limelight when it comes to research and conservation efforts, and with good reason. These charismatic species, such as wild dogs, cheetahs, lions, even leopards, have a core conservation and research focus in many areas. But when it comes to on-the-ground management, we need to go beyond the charismatic. Interspecies interactions are complex and affect how we manage systems. But how do we consider these interactions if we're not sure how many of these species even occur in these areas? One spotted hyena in an area or a fenced reserve is going to have vastly different effects than a complex multi-clan system. So what do we do? Fortunately, in conservation areas, there are a lot of different research projects. These research projects often come with a lot of bycatch data. Research projects are often species specific. This means their survey designs are set around targeted species or around their objectives. Despite being species specific, they still catch information on all the other species that exist. In conservation areas, oops, jumping ahead. So there's, anyway, species specific designs. Conservation areas again, as we say, have a lot of other species which we need to focus on. These conservation areas host a variety of different research projects, and although they may be species specific, again, they have different bycatch data. Madikwe Game Reserve is an example of a site that has hosted multiple different research projects. These projects had their own objectives and again, own designs. The projects ran over different survey lengths. Panthera leopard monitoring is a project which runs annual leopard surveys, or annual camera trap surveys, which target leopard population trends over time. Their surveys tend to run for about 40 days annually and then are removed. There's the Snapshot Safari project, which also uses camera traps to monitor biodiversity. These cameras are set up and tend to run over the span of years, depending on what the site needs or allows. We also ran targeted surveys, which were directed more towards cryptic carnivores. Our surveys were baited and moved sequentially throughout a reserve over an 8-month period. So you can hear from these descriptions, these surveys are all very different, with very different designs and ran over different timescales. During this time, Madikwe had specific questions about which carnivores they needed surveyed. These were brown hyenas, spotted hyenas, and leopards. Analyzing these surveys alone produced relatively low capture probabilities for these species, which is a common problem for nocturnal and elusive species. So why not combine these surveys, use this data collectively to produce a huge amount of data? Well, one of the major problems when doing this is statistical models are generally designed for more structured surveys, and especially in population models, they're designed for surveys which run over shorter periods of time. When you start combining a lot of different data, you start increasing the number of variables you have to consider in these models. So after considering multiple variables, one of the major factors which affected our models was the extreme fluctuation of camera traps at any given time. Camera traps ranged anywhere from 56 cameras up to 150 cameras over the study period. We adapted Bayesian capture-recapture models and developed a model which could account for these fluctuations. And thus we're able to produce robust population estimates for Madikwe Game Reserve. Now we focused only on Madikwe, but multiple reserves have different research projects. So the extensions exist beyond this. We can even go beyond research projects and look into anti-poaching cameras. A camera trap set up for security is still catching data on everything moving by. So there's a wealth of data available. So if you can catch some poor unsuspecting student to process all of this data, the applications are extensive. I'd just like to say thank you to our contributors, which is Oppenheimer Memorial Trust and Northwest Parks Board, as well as Panthera and Snapshot for sharing their data. Thanks.
Thanks, Terry. Yeah, camera traps is definitely becoming more popular, so there's a lot of data out there. So I'm not going to allow any questions. If you guys want to chat to her, please do so at the poster session. So then we're going to move straight on to Corin, who's going to be talking about the use of artificial intelligence in conservation ecology. Thanks.
Thank you very much. And for those of you who don't know me, I'm Karen. I'm from Rhodes University. I'm not an ecologist. I'm a computer scientist, so I'm not going to hurt any animals or plants in this presentation. My title is slightly different to what you might have on your timetable, but it's really deep learning that I want to talk about. This deep learning is just really an aspect of artificial intelligence, and it's hoping to give you some insight into how you can use this technology or these algorithms, because they really are just algorithms, in kinds of studies that you might be wanting to do. So before I give you some ideas, I'm just going to give you a little bit of talk about what is this concept of machine learning. And it's a very old concept. It started out as being a mathematical computational model to try and mimic the brain. So in the brain we've got neurons, we've got synapse links between the neurons, and this is how we learn as humans. And the computational model that was set up was the artificial neural network, and it also had neurons, which we call nodes in computer speak, and it also had links linking up the nodes together. And the important part here is that our links have weights associated with them, and that is to show the different a strength or the strength of the influence that each node has on the next node. And this is just a diagram of a very simplistic artificial neural network where you have at least sort of the input layer is always got to be there. That's where your external data gets input into. And the external data can be anything from numeric data, it could be images, it could be acoustic data, and that's the data that has the features in it. Now, we use the word features a lot when we're talking machine learning, but it's just really like how we learn. So a feature to us, if we were trying to recognize another colleague, for example, the features that we might use would be the shape of their nose, their eye color, maybe their hair color, maybe the shape of the chin. And maybe after my talk today, you'll all start forgiving yourselves for having not recognized your colleagues when they were wearing masks. And that's because maybe the features that you used to recognize the colleague were hidden by the mask. So it's not a problem. So the input layer is then where the input data comes in. The magic happens in the middle, the hidden layers. This is where the processing happens. And remember, this is just a mathematical it's a mathematical model. So the computation that's happening is really just some mathematical equation. And then at the output layer, that's where we get the answer that this neural network has been set up to compute. And what happens in that middle? What is the magic? Well, the magic is, as I said, a mathematical equation. We typically take the inputs that are coming in. We get a weighted sum of all the weights of the inputs coming into a node. We add some kind of bias. We send it through some kind of activation function, which is typically just a nonlinear function, something like sigmoid. And we then get our output. But this is far too simplistic. The real magic and the real power of the neural network comes from the learning ability. So the network itself has the ability to learn where it's gone wrong. And the way that happens is that when you get the output from the network, if the output doesn't match what was expected, then we go back into the nodes, into the links, and we change the weights. Well, we don't do it, the network does it. It's all done automagically, which is what these networks are why they are so powerful. Pretty much after multiple iterations, which we call epochs, and these epochs could run into the thousands of iterations, your network might have the weights that have been set up correctly so that when you put something in, it comes out with the right output that was expected. Now, I know this all sounds like it's far too much like magic and it can't be true, but the power or the real magic comes from this learning ability, the ability of this network to learn. And there's a number of different learning types or types of learning that the networks can be programmed to use. The reinforcement learning, which is the last one on the slide, is something I'm not going to talk about because that's really not applicable to ecological studies. That's something that might appear in, for example, a computer program that's been programmed to play chess, and where it gets a good reward if it makes a good move like taking the opponent's queen. The two that I am going to talk about are the supervised learning and the unsupervised learning. So the supervised learning is like having a teacher. You get feedback all the time. The network knows whether it's got something right or wrong straight away. And the kinds of tasks that a supervised learning system can do is classification tasks and regression tasks. The goal is then to predict the behavior or to make a decision based on past data that's been given. It's a wonderful learning system, but unfortunately the challenge to use this supervised learning is that you need plenty of data to train with, and the data has to be labeled. In other words, the data has to be both the data plus what the outcome should be. The other learning type is the unsupervised learning, and there the machine is trained with unlabeled data, so it's a lot easier to train. Obviously, you've also got to give some kind of cost function that can map from the inputs to the outputs. But the types of tasks that you can do with unsupervised learning are far reduced to those that you can do with supervised learning. So unsupervised learning is really good for exploring big sets of data, finding patterns, doing some clustering, maybe doing some filtering as well. And then you can determine how accurate your model is based on how similar your elements in the cluster are and how dissimilar each cluster is to each other cluster. The advantage is obviously your training data is easier to create, but as I said, it's limited in its application areas. So this is just a diagram of how you would, what a typical machine learning system would look like. And you'd have your training data here. You'd either choose the unsupervised or the supervised learning approach. You'd shift it into some kind of feature extraction. And in the traditional machine learning, the feature extraction had to be done by the human, the person that was running this. And the feature extraction would be to kind of identify those features in the data that the network should concentrate on. In deep learning, you'll be very pleased to hear that both the feature extraction and the machine learning algorithm gets done automatically by the deep learning network. So it's no longer the part of the human to say, only look at these features in the data. And as I said, the features in the data could be parts of an image or specific areas in numerical data. And then what you get out at the end is either some kind of grouping of objects, in other words, a classification task, or some kind of predictive model in your, from your data. What I'm going to do next is I'm going to show you 3 separate research projects that use different variations of these supervised and unsupervised, and just show you what's possible. So this is a study that was done at Rhodes by one of our master's students, and the aim was to be able to show this network, this trained network, images of leaves, and for the network to be able to classify what the plant was that it came from. Obviously, it could only classify plants that it had already been trained with, and also to show if the leaf was diseased or not. So here you've got images of a diseased leaf and a not diseased leaf. And what was used here was a convolutional neural network, which is a great network for using with images. And the result at the end of the day was that the accuracy that we got was somewhere between 91.25% and 95%. And the difference there was that the 91% was done on a very small training data set, and the 95 was when we did it on 2 data sets that we combined, and the accuracy improved because the network had been trained on more examples. This is a thing called a confusion matrix. And this is kind of to show you what the results were of that study classifying leaves. And what you've got along the x-axis is the true label of the images that were tested. So what they should have been. And you've got things like lemon healthy and lemon unhealthy. So we had lemon leaves and guava healthy leaf and a guava unhealthy leaf. And along the y-axis here, we've got what the model predicted. So if the model predicted what the actual true label was, this is all these numbers down the middle here. So mostly it predicted things correctly, both whether it was diseased and which plant species it came from. This was done on a 20-class model, so there were 10 different species of plants, each having diseased and undiseased. Diseased and not diseased. There were also some places here where you can see there's 4 times where the lemon healthy leaf was classified as unhealthy. The second study that I can show you is about detecting the Hackia species from drone imagery. And in this example, we wanted to not just detect if the Hackia was present in an image, but we wanted the network model to segment it, to actually give us a picture of where it was. So the image at the top has a Hackia species in, this image at the bottom has just 2 shrubs that aren't Hackias. And we ended up with results that looked a bit like that. These were the images that were sent in, and we ran it with a number of different tweakings to our model, just to show you that you don't always get the model right the first time. You sometimes have to tweak things to get better results. And from here you can see that we actually found the Hackias, and this is how the model looked at that and actually could segment the area that the Hackia was in. This is a study that wasn't done at Rhodes, but it's a great example of unsupervised learning. Both the previous two were supervised. This is unsupervised. And this was a study done by Sonnewalt et al., and they wanted to determine global marine ecological provinces based on plankton community structures. And they had 55-dimensional data that they started with. They sent it through some kind of dimensionality reduction algorithm, which is also part of the machine learning sort of packages of algorithms. And they came up with 11-dimensional data. This was biomass of 7 functional groups of plankton and 4 nutrient supply rates was their data. And after the unsupervised algorithm had gone through it, it managed to classify regions in the oceans into 115 different clusters. And what this is the output of their study, and it shows the different colors. The bottom here from 0 to 115 are each of the different clusters that were obtained based on the similarities of the plankton data that was fed into it. So this is an example of an unsupervised clustering model. There's many, many other applications of machine learning. There's a wonderful review paper by Kristen et al. Of 2019, and they come up with all kinds of different things that you can do. You can identify and classify animals or plants based on images. You can identify birds or mammals or bats from acoustic data. You can do phenotyping, in other words, counting leaves to assess the growth of a plant. Animal behavior. You can look at animal trapped images, and you can decide, or the model can be used to decide whether the animal was feeding or sleeping or whatever they were doing in the images. Population monitoring, so for example, detecting tree defoliation or diseases in crops or scars on animals. You can get pictures of animals and detect if they're healthy or not based on how many scars or they've got. You can do prediction aspects or tasks from whether there's going to be an outbreak of a disease based on historic data. Pity we didn't have that before COVID. And several others. Landscape analysis for large-scale monitoring. You can have images, and you can analyze when the cotton fields are in bud based on the images that you get for that. I'm going to just move on. The same review that I've just talked about that came up with all these different options of usage also did some literature surveys to determine how many ecological studies were actually using deep learning. And they did the survey in 2018, and with certain keywords like ecology plus a whole number of these, they found that there were only 87 papers at the end of 2018, well, in fact, a couple also in 2019 there, that had actually made use of this new concept. So what I'm really proposing is that obviously the number has increased because we've had 2019, a couple of years in between, but the reason I came here was to really kind of almost say, think about whether it's possible in your own research, whether you can actually make use of some of these machine learning techniques, specifically the deep learning ones, to actually help you assess your data. And the same Kristen et al. Came up with kind of a flowchart of how to decide whether you can use it or not. And they start off with the raw data, and the first question is, do you have a lot of data? And if you answer no, then clearly machine learning is not going to be for you, and you must go and use some other learning approaches or just some statistical techniques. If you answer yes, then if you want to just explore your data, you want to just see what filters stuff or maybe classify or see similarities, then you would go the route here of doing some unsupervised learning models. If you want to classify, detect objects, or identify aspects in images or in data, then you would go the supervised learning route, and that would then involve you creating a model, which would mean that you must first decide on an architecture. There's a few classic deep learning architectures around. And you also then have to decide on what framework you're going to use to actually program it. And I always advise scikit-learn as being one of the simplest packages out there. It's a Python-based package or Python-based library for machine learning. Once you've got your model, you then have to train your model, and we typically use between 70 and 80% of our training, of our data that we've got available to train the model, and then we use the remainder, the 30 or 20%, 20 or 30% to validate the model, because there's no point training a model and then never checking that it actually does what it's supposed to do. There's always, unfortunately, once you've got the trained model and you've checked the accuracy, you then can determine whether your results are satisfying enough. In other words, is the accuracy acceptable for the task that you've got that you've created the model to achieve? If it's not yet the accuracy correct, then there's quite a lot of parameters that can be tweaked. Different loss functions can be used. I haven't mentioned that before, but that's typically the one that we used for our Hackier study. We changed the loss function and we got a way better model. And you can go back, refine the model, and then go again. And once your results are accurate enough for the task that you have, you can then use the model to do the task. And the auto magic happens for you. And that's all I've got for you this afternoon. I'm happy to talk to anyone afterwards in the poster session. Thank you.
Thanks, Corin. So with all this camera trap data and the volumes of data that's exploding, I definitely do think that some of these machine learning options need to be pursued. So if anyone's interested, talk to Corin at the poster session. I do hope that you have a couple of spare students that could possibly get involved, because I think you might need that. Okay, so we're going to move on. Our next speaker is Albert, and he's going to tell us about using UAVs in the Olifants River Gorge. Thanks, Albert.
Oh, thank you. Very interested in that AI sort of machine learning talk, because I've got thousands of images of crocodiles at this stage. So yeah, for those of you who attended yesterday and didn't slip out and skip the session, you'll know why the Olifants River Gorge is quite an important place for crocodiles. It was the site of a mass mortality event for crocodiles in 2008 to 2010. And since then, there have been a few attempts to sort of get an idea of what the size and the structure of the crocodile population in this area is. Research in this area is dated back to like 2000 and even before that, when Swanepoel et al. Was looking at the breeding territory in this area and the importance of this area as sort of a breeding and maturing reach for crocodiles. So this work was done in collaboration with the Davies lab from Harvard. They flew the drone, and I was lucky enough to get all the thousands of images, and I had to go and just count the crocodiles. So that's what I'll be presenting for you today. So that's the Olifants River Gorge. It's quite a deep gorge where it goes from the confluence between the Olifants and the Letaba rivers down here. And then you can see this is an elevation model that just shows the gorge as it stretches towards the mouth of Lake Mazenga there in the northeast. The problem with traditional survey methods is that they are manned platforms, so they are subject to observer bias. You can imagine someone hanging out the side of a helicopter shouting 3 large, 1 medium, and so on, and that's how crocodiles are counted. So they have relatively low resolution and large errors. Something that I didn't put here, which is also important, is that they have quite a high level of disturbance. When you fly over those crocodiles, they'll scatter into the water, making them quite difficult to count. So UAVs really offer this repeatable platform that you can fly the same flight paths over and over again because they're autonomous, and they produce high accuracy rectified aerial images, and they have relatively low disturbance and crucially no observer bias. So just a very broad illustration of how this works is you take an area, you'd send your UAV on zigzag paths around the site, you take thousands of images, and then you'd stitch those together to create orthophoto maps of a site. If you do this over large enough areas, before I move on to that, that's just the resolution. So you can see easily identified crocodiles on this little beach there on the border between South Africa and Mozambique here. If you do this over large areas, you can get we covered the entirety of the Olifants River Gorge, so we've got orthophoto maps stretching the whole thing, and we could count all the crocodiles. Just some nice images here of relatively large crocodiles with cohorts around them, and we suspect these are large males that have maintained some breeding territories. So using this method, crocodiles are relatively easy to count, and I could produce a map in the sort of traditional survey reaches and compare that to some manned aerial counts that were done just a few weeks prior. And you can see the discrepancy between a fixed wing manned count and that of the UAV, almost 4 times more crocodiles counted using a UAV than a manned count. But to measure the crocodiles is something different. So here we want to look at distinguishable morphological features. And if you look at, I've just taken some of my crocodile farm work on the right here to just show the discrepancy between orthophotos at 2 centimeter resolution versus those at sort of 3 to 5 centimeter resolution. You can really see the difficulty in distinguishing morphological features. When you measure crocodiles, you're looking at something like snout vent length, which is the third circumcircle scute layer behind the back legs, and you can't see that in images like these, even if the crocodile is out on dry land. So I used some of my crocodile farm data, and there I've got thousands of crocodiles to develop just a relationship between distinguishable morphological features that you can see from aerial images. So something like the head to the end of the neck, which is differentiated by the front limbs, and the head to the end of the hind limbs, and I could derive relationships between those, and that enables me to measure crocodiles from relatively lower resolution aerial images. So those are just the relationships there. Applying this to the crocodiles in the Olifants River Gorge, I could measure almost 80% of the crocodiles in the gorge, and so I could get a really nice size distribution in the gorge, and we see a lot of crocodiles in the sort of 3 meter range of length, and then some very large individuals over 4.5 meters. And this is novel data, this has never been done before. This allows me also to overlay some size and count density. So this is just a kernel density map, and you can see crocodiles congregating in several areas, and those arrows just show you where there are crocodiles larger than 4 meters. So you can see a nice distribution, which suggests maybe that this is a breeding area where males maintain breeding territories. That's just a little example of crocodiles congregating in an area there. This also allows me to use some previous studies to estimate biomass, and we could work out that there's almost 35 tons of crocodiles within this 8-kilometer stretch of river, which equates to about 345 kilograms of crocodiles per hectare of water, we use that as a substantiation to show that crocodiles really do not rely just on fish, because you cannot support almost 400 kilograms of crocodile with just a hectare of water. Thank you very much.
Thanks, Albert. So we're going to move on to the last talk in the session from Nicholas, and he's also going to look at some photogrammetric configurations for measuring animals. Thank you.
Good afternoon, everyone, and thank you for the introduction. So I'm going to be presenting an application in zoology that was born out of the hard lockdown period in South Africa. And we, as photogrammetrists working as part of photovolumetrics, we needed to be able to access our sites, but we weren't able to do so. So what we decided was that we would bring those sites to us. 3D close-range photogrammetry can employ multiple perspectives to reconstruct an animal in 3D space. And if we're looking at living, breathing, moving animals, one of the most non-invasive ways of doing that is if we have those multiple perspectives synchronized to take photos simultaneously. These can be triggered through the motion detection event, either through an infrared sensor or via a substantive change in an image. And these can then trigger those synchronous photographs to 3D reconstruct that animal. This over here is not a scene from Star Wars. This is one of the first photogrammetry stations. It was applied in Tswalu. And it's basically a gazebo with multiple cameras pointed inwards. There's an infrared motion sensor. If that motion sensor is triggered, multiple cameras take photos simultaneously of the animal within the scene, and we can then 3D reconstruct that animal as presented in the bottom left corner there. And there are various approaches that have followed on from this method. We have one such station sited at the Nwasichaka watering hole, and it's been there since 2015. It is 30 minutes out from Skukuza and 4 to 6 hours from our home base in Pretoria. It consists of 10 cameras, all pointed inwards, some positioned high on marula trees, some positioned low in bushes and barricaded against various animals. And there is a motion detection camera that then sends a signal to a brain, which gives a command to the rest of the cameras to take photos simultaneously, and in that way we can photogrammetrize any animals passing through that area. It is completely off-grid, and there is a solar farm specifically for the site, allowing us to keep it powered. Despite it looking, and although it looks very idyllic, it does have its challenges. So this site has gone through multiple iterations over time during its development, changes in cameras, in their models, in their spacing, as well as various changes to the solar power associated with the setup. And there's also been some maintenance associated with the destruction by the elements and by animals pulling cameras off trees and pushing them over. So you can imagine with the multiple iterations that's required for this trial and error approach to developing camera configurations, that there's a lot of time and finance associated with such a development of a remote configuration. This brings us to virtual photogrammetry, wherein we are able to simulate photogrammetry within a digital 3D scene. And it is well established in technical fields where the relationship between virtual photogrammetry and real photogrammetry has somewhat been established. This is complicated a bit further with the use of animal models, where there's additional complexity associated with this complex form and movement. So we aim to develop a framework where we can design all our photogrammetry configurations within this digital 3D space, regardless of the scenario. So we were testing various scenarios, but today I'm just going to present the volume and the bird flight height. We are going to, or what I'm going to do is I'm going to demonstratively determine whether the top-down camera, an additional top-down camera at that Nwasichaka site that I've just presented to you, would improve the accuracy of that photogrammetric configuration. And then for the bird flight height configurations, which is a stereo photogrammetric configuration, so that's just 3 cameras pointed down a field of view, whether the accuracy in that declines with distance. We come to calibration. Calibration is usually a non-topic when it comes to photogrammetry. It's usually just a brief statement in your manuscript. But when it comes to virtual photogrammetry, we need to talk about it a bit more, because we need to calibrate those real cameras and then extract those calibration parameters and employ those in our virtual cameras so that we can appropriately simulate those cameras within a virtual setting. And those are just some of the parameters that we've used in that. If we want to produce the 3D scene, we've got multiple options in doing that. We can simply 3D model it. If we had the opportunity, the ideal scenario would be to actually 3D reconstruct all of the objects within the scene, especially some of the more prominent ones, 3D reconstruct the ground plane. But I'm going to present the non-ideal situation where you are just presented with, the 10 photos that are available to you on your little SD card. And you can basically just do a normal project as you would, as if there was an animal within that scene, manually mark, include the scale within the scene, so you've scaled that scene within that 3D space. Manually and mark and cross-reference your cameras so that you can place those cameras within the virtual setting. And then just do a smart match point cloud generation, just for some extra detail, and triangulate polygons between those points. We can then import that scene into Blender and then overlay the photographs, the original photographs on the virtual cameras. We can position those cameras at the points that were cross-referenced in the original photogrammetry project. But those cameras do not have an orientation associated with them. So overlaying the images actually allows us to align certain features within the photographs and the photogrammetrically reconstructed scene. And in that way, we can reconstruct the orientation of those cameras as well. We can model some objects within the scene. So many of the objects are near the edge of the scene, so there's only stereophotogrammetric information about them, just simple linear measurements. And then we can also use a reverse silhouette approach to basically map out where those objects occur within those fields of view. And these include trees and logs and some rocks and stuff like that. And we can also just a triangulated ground plane between some of the manual points that are placed across the original project. We import animal models. These animal models can come in the form of a digital 3D repository, or they can come from photogrammetrized animals that have been taxidermied. And it's up to you. It's important that we texture both the 3D scene and the animal models. Otherwise, you're going to end up seeing pink elephants. And then we need to realistically simulate the lighting, and we do this through a black body node, which allows us to change the color of this directional sunlight. So we are able to adjust both the intensity and the color of the light depending on that angle of incidence from that light. We need to be able to simulate the photographs as well. So we add a number of the settings that we get from the calibrations. We can add a lot of the additional settings, such as shutter speed. So if you've got an animated model moving through your scene, you might have some motion blur within that scene as well. And we can also affect the depth of field with an aperture that you can assign in this nifty little photographer add-on in Blender. We then add a distortion to this scene. So there is lens distortion associated with photographs, so we have to reintroduce that lens distortion, and we get those distortion parameters from the original calibration. We reintroduce it, and we can see those edges kind of pulling in with that introduced distortion. And then we modify the EXIF headers of the images so that the photogrammetry software can read those images as photographs. We're basically tricking the software. And then we would apply the project as we would for the real scenario. In this case, the real scenario was a silhouette volume photogrammetry project. We do some manual landmarking. We process for orientation, so we can see those cameras orientating as they would for a normal project. And then we do a silhouette of that animal, and we can actually employ some of the segmentation techniques that Karen mentioned earlier. And then we cross-reference those silhouettes to then produce a 3D volume within that scene. And I would like to talk about the elephant in the room here first. One of the cameras was actually pointed down too low in the original images, and there is a recommendation that we can adjust that camera to be pointed more upwards. Now, I don't have to go to the scene and actually adjust that camera upwards. I can just rotate that camera slightly upwards within the virtual setting, test whether that actually improves the model, and then go forward with that. So there are so these are the models that result from the photogrammetry. The reference, so the known dimensions of the volume of the animal model that were introduced were used as a reference, and accuracies were then determined from that reference. And we can see that the adjustment of camera 2 greatly improved the model, or improved the model slightly, but the addition of a top-down camera improved the model significantly because we had the removal and the cookie cutting of those ears away from behind the ears and in front of the ears as well. So we improved the silhouette from the top of the model, and we cut away from that additional volume that's integrated into it. And we can compare this in Cloud Compare as well. We can identify areas on the surface that have that high degree of error, and we can see that the addition of that top-down camera removed error behind the ears and in front of the ears as well. But there are still some suggestions. We could actually add some additional diagonal camera angles, for instance, to remove that additional volume on the edges there. We did the same for Impala, and here there was a slight improvement with the addition of a top-down camera. And we can see the red on the error of the surface, and we can actually add an additional diagonal camera angle to try and remove that as well. So this was similarly applied to bird flight height, and I'm not going to go through all the methods of that, but we have some preliminary results from that showing that there is an increase in, or there is a decrease in accuracy with an increase in distance away from the camera, and this is usually associated with the pixelation. So images rendered from Blender have the same pixelation as images that are photographed. So there is this greater uncertainty the further the bird is away from the camera in trying to pinpoint its position. And there is some evidence showing that an angle of intersection of 15 degrees is optimal, but we need more data for this to actually for it to be more conclusive. So this is an ongoing research area, and we're still trying to improve the virtual representation of real photogrammetric accuracy. We have done some tests where we've shown that the actual measurement accuracy does correspond quite well with the virtual measurement accuracy when it comes to volume of a point cloud. So a point cloud is a lot more accurate using more cameras. In this case, it's about 24 cameras, and you can produce them in that we're giving these students free cameras in a virtual setting. There's minimal risk because we're not getting an animal involved, and there's a lot of ethical implications that are avoided in that case. But they still get the same experience of being able to design a camera configuration. So given that virtual corresponds to real, this gives us the ability to test the accuracy of photogrammetry configurations, which may sometimes be infeasible. If we're thinking of the volume of an animal, one of the most accurate ways of getting the volume of an animal is actually to dip it and get its displacement volume. And there's a lot of ethical implications there as well. Or we may just want to test an approach before actualization. So let's say we've got a completely novel photogrammetric approach that has not yet been applied. It may benefit us to actually test it in this virtual setting first before spending so much time and money before we actually apply it. And it is a versatile framework. We're testing it on a number of scenarios, and we hope they pan out. I'd like to thank the following parties for their contributions. Are there any questions?
Thanks, Nicholas. So it looks like between your cameras, the drones, and Karen's neural networks, you guys aren't going to be getting into the field as much. So if there's time for one quick question, if there is. Nothing going. Well, thank you very much. So please see Nicholas in the poster session if you have questions. And then thanks to everybody for the last session. And please come and enjoy a glass of wine and some snacks and the posters. Thank you.